All right, so we've learned about sex linkage. Now I'm going to tell you a little bit about a different type of linkage called we're just going to call it what the books call it, just gene linkage. Gene linkage. And how is that different from sex linkage? Well, this is when genes for two or more, can't spell, different traits are located on the same autosomal, that means not sex chromosome, but one of the other 22, autosomal chromosome. An autosomal chromosome is a chromosome that is not a sex chromosome. That's all it is. So one of the other 22, right? And you can have, I mean, obviously, that shouldn't be that mysterious, right? Because now we know that there are thousands of genes located on a single chromosome. So obviously, many genes are linked onto the same chromosome. We know this. Mendel didn't know this, though. So he very luckily studied pea plants, and he studied traits that ostensibly each one of the traits he studied, like plant height and seed coat, must have been located on different chromosomes, because if they hadn't been, then his Punnett squares wouldn't have worked out so mathematically perfectly for him. Because remember those three laws he came up with? Law of dominance, law of segregation, law of independent assortment, especially that last one, the law of independent assortment. Genes can't independently assort if they're stuck on the same chromosome. We know that now because you've studied meiosis in detail. Gregor Mendel didn't know that, and luckily he didn't have to deal with it. Okay, so remember that these would be traits that cannot follow the law of independent assortment. Independent assortment. And again, I think it's much easier to draw a diagram, an example for this, to show you what I mean by that. So let's say, for example, that we have on one side of the screen, I'm going to split the screen. On one side of the screen, I'm going to show you an example of non-linked, non-linked dihybrid cross. Dihybrid cross, right? And over here, I'm going to show you a linked dihybrid cross. There we go. Okay. So let's just take the single organism, and it's making it has chromosomes. If they are not linked, right? Then those chromosomes, let's make two homologous pairs here. Because what's going on is you have, get some colors here. All right. With non-linked, you would have this situation. You'd have the alleles for these two different traits located on two different chromosomes, right? They're on different chromosome pairs. Whereas over here, chromosomes for one person that's about to undergo meiosis and make their gametes. Those A and B alleles are stuck on the same chromosome. So you'd have this. Maybe you'd still have your A's and dominant recessive there, but maybe your B's would be down here on the same chromatids. There. That's the difference. The non-linked are going to be riding on different chromosomes. Therefore, when meiosis comes along, remember the left side is non-linked. These guys can independently assort. You can change the position of those chromosomes across the metaphase plate during prophase 1, and that's going to result in, remember this picture? When you're figuring out the gametes to go on your Punnett square, and we did this, each type of A goes with each type of B, right? Each type of A can independently assort with each type of B, and you can therefore get all 4 different types of gametes equally, right? You could get an AB as often as you could get a little a big B. You could get a, right, am I doing that right? Yes. Oops, okay. I didn't exactly do it the way I usually do it. There, okay. 4 different types of gametes, all equally plentiful, right? All equally plentiful, and therefore when you set up the dihybrid cross for this in that giant Punnett square that I'm not going to do here, your dihybrid cross is going to be a 9, 3, 3, 1 outcome, which, by the way, you can always assume as long as there's no linkage and as long as both parents are complete hybrids like these, you should always get a 9, 3, 3, 1 outcome. That tells you, if that's what your offspring actually fit, that outcome, that there is no linkage. Now go back up here, though, and let's look at this linked issue here again, because that's going to be different. These guys cannot. They can't just switch places and change positions. They can't independently assort because they're stuck on the same chromosomes. The A's and B's are stuck on that same chromatid, right? So this guy here, A stuck with big B, A stuck with big B, little a stuck with little b, little a stuck with little b, right? So they're going to carry those back and forth, and you can't mix and match them. Right. So what does that look like? It looks like this. The big A is stuck with a little b. No foil arrows here. And the little a is stuck with the little b. And so really the only gametes that can come out of this are big A, big B, and little a, little b. Because they're stuck together, right? So the results will not be 9, 3, 3, 1. There'll be something different. Now, the only way, because sometimes you do get a few of these. You get a few of these. Let me just write it down. The only way you can get, and they call these recombinant gametes, right? Recombinant gametes, which would be little a, big B, or big A, little b, right? The only way to get that is how? I bet you can guess. How could you get those if the alleles are writing on the same chromosome? I'm going to draw it for you. Bet you'll get it before I finish drawing it. Okay. We still have the big A's up here. And the little A's are still staying in the same place. And this is still A, big B. Yes, you should be seeing it crossing over, right? Here comes the little b exchanging its place with the big B. And that's a crossover event. Crossover events can exchange some of the alleles with the others and therefore give you either one of these or one of these in the gametes, right? But the crossover frequency, and this should make sense to you, is going to depend on how far apart the A genes are from the B genes. The further they are from each other, the more likely there's going to be lots of recombinant gametes because there's going to be crossover events and they're going to be able to change places because they're far away from the other genes, right? But if they're really close together, right? If like this B was here. I'm going to take you to a pear deck in a second. So we'll, all right, if you had, instead of that configuration, if you still had linked genes, but, you know, here's your A's and here's your B's right there, locations right there. So here's your little A and here's your little B, right? And if that's the case, the crossover frequency is going to be much less because there's not a lot of distance between the genes. And the likelihood of this chromatid crossing over right between there and those getting exchanged is less, right, than over here where you have all this distance between the A and the B, and you have plenty of places for crossover to happen and to switch those genes places. Okay, so that's the difference between genes being linked and genes not being linked. Okay, so I'm going to stop this recording now.